Okay, let's get started. We are looking at developing new drugs, which starts on page 241 of your textbooks. As you can see from your LI, you guys have to have an idea of the historic drug testing protocols, specifically the work of William Withering and Digitalis Soup. So to give you guys some idea of the time period that we are talking about, Withering's birth and death dates 1741 to 1799, and Withering actually did some work with Digitalis. So Digitalis is a chemical found in foxglove, which is a flowering plant found in the UK and Europe. There is a picture of one on page 241 in your book. I would recommend having a look and being able to recognise it in case just a picture comes up in the exam situation, you'll be able to see what the plant is. But these foxgloves are flowering plants. They're found in the UK and Europe, and they contain a chemical compound called digitalis, and this actually affects the beating of the heart. So for centuries it was used as a poison, so we could use it to kill people because of that effect that it has. However, during this time period there were also many reports of foxgloves being used to cure the dropsy. So dropsy is was the term for what we would now call edema, which is the swelling of tissue due to fluid retention, and it's commonly seen in people who have heart failure. So failing circulatory system, quite commonly you get this edema, this swelling. So in 1775, a patient came to see Withering. He was a doctor at the time, and they presented with a serious heart condition. Withering couldn't give this patient any treatment, so he sent her off to a wise woman. So a wise woman would have been sort of a local herbal healer, so they would have used sort of natural remedies, plants, herbs, spices, things they could get their hands on. And anyway, this wise woman gave this patient a soup, and Withering noticed that his patient actually got better after drinking this soup. So he bought the recipe for it off of this wise woman, and there were about 20 or so ingredients in it, and when he looked through that list, he guessed that foxglove contained that active ingredient. It contained the component that was helping his patient with her symptoms. So over the next 10 years, he tested a variety of potions made from foxglove. So he prepared them in different ways, he prepared them in different concentrations, gave different amounts, etc. And over the 10 years, he tested 163 patients, and he gave them this potion and used it as a treatment for dropsy whilst working at Birmingham Hospital in the UK. Many of his patients did get better, but some of them almost died of digitalis poisoning. So those that had complications tended to find they suffered from nausea and vomiting. So he obviously found out some of the side effects of digitalis. And those that were cured were cured because they had increased urine output, so that helped to remove the excess fluid. And he also noted that their heartbeat returned to normal in those patients. So whilst we call it a soup, he was actually making this soup, Withering was making this soup from dried and powdered leaves of the foxglove. So he found that boiling the leaves reduced the effect of the drug, so it was helping to minimise those side effects. And actually in doing this procedure, he was extracting the digitalis chemical. So much like we spoke about extracting and purifying compounds from plants when we talked about plant-based medicines, he was doing sort of a more low-tech version of that. And today, there are drugs that are based on those chemicals, based on that digitalis from the foxglove, still in regular use. Digoxin, for example, heart-based medicine. So there are loads of prescriptions out there that would contain that active ingredient still. So obviously, when Withering was doing his work, there were not any rigorous testing protocols, but nowadays we have considerably more rigorous testing. So bear with me 1 second, just get to the right page. There we go. So every new medicine that we hand out today is a result of years of research and development. So it's not just randomly given out to someone. It has to go through thorough testing, and in order to sort of be approved for the mass market, new medicines have to be effective. So it has to cure, prevent, or relieve symptoms of the disease that you are expecting it to. So it has to actually work. It has to be safe, so it's got to be non-toxic, and the side effects have got to be acceptable, so sort of minimal side effects. It's got to be stable, so you've got to be able to keep it and store it under normal conditions, for a relatively long period of time. It's got to be easily taken into and removed from your body. So it's got to get to its target organ or its target location with ease, and you've got to be able to remove it from the body once it has done what it needs to do. So it can be excreted, and you need to be able to make them on a large scale. So that just in terms of being able to produce enough to meet demands. In terms of producing and researching and developing a new drug, it takes around about 10 years from first sort of discovering a plant through to your doctor writing your prescription for it, and around about $2.6 million to develop a new drug. So it's a really costly and quite lengthy process. Now, the testing that you have to go through does vary from country to country. So each regulatory body might have a different testing regime. But generally speaking, you can go through sort of 6 phases of testing. Cell cultures, tissue cultures and organs. So as their name suggests, you start off small, testing it on cells, up to tissues, up to organs. So you might be testing it, if it's a heart medicine, you would obviously test it on a heart to see what happens. Those 3 stages come under the sort of research umbrella. We then move on to the development side, and that's where we do animal testing and clinical trials, phase 1, phase 2 and phase 3. All of those phases are slightly different. You guys need to have an idea of what's involved in these 3 stages and sort of the advantages and disadvantages. So that's what we'll look at in a moment. But as you can see, at the very beginning, you might start out with sort of 10,000 different chemical compounds, or 10,000 new suggested medicines. And by the end, when we're doing our clinical trials, you sort of whittle down to 5. In these first 3 stages, there are lots and lots of failures, thousands of failures, because it's discovered that it has a negative effect on tissues or perhaps an organ, or it just doesn't do what you actually need it to do when you get up to a whole organ level. All right, so now flicking back to our different types of testing. First one is animal testing. And this is used to find out how it works in a whole organism. So up until this point, you might have tested it on an organ, but you need to test it to see how it works when it's in an entire sort of living, breathing thing with different organ systems, different enzymes, different chemical reactions going on. So when you're looking at it at an animal testing level, you're checking your delivery system. So is it a tablet? Is it a liquid medicine that you swallow? Is it an injection? Is it a nasal spray? You need to test how you're going to get it to the target within the body. You're testing its stability, so to make sure it's not going to break down into something toxic, or it's not going to be broken down sort of by enzymes and chemical reactions in your body to make it inactive before it can work. And you're also just checking to see if it's changed chemically, is it excreted safely, kind of looking at it from a whole animal, whole organism perspective. We use mammals because they are as similar as possible to humans, so things like mice and rats. Those are chosen because they're smaller, so they're easier to care for humanely than, say, a dog, and also because people tend to find testing on mice and rats slightly more agreeable, slightly more palatable than the thought of testing on a cat or a dog, or a larger, like an ape, for example. However, some testing does require that you carry it out in 2 species, both a rodent and a non-rodent, so one in a rat or a mouse and one in something else, so they're not the only possible animal that you might choose for your animal testing. Some negatives associated with animal testing. The obvious is obviously ethical objections. People do not like it, they do not agree with it. It's also expensive and it is time consuming. So because of these reasons, animals are starting to be replaced either by tissue cultures or computer models. We're also trying to minimise, as scientists, the number of animals that we actually do it on, and we have to make sure that any tests we are doing are, the protocol is designed as such as to minimise distress. However, these tissue cultures and these computer models don't provide enough information yet for you to be able to move on to clinical trials. So whilst they exist more as a way to minimise the amount of animals or number of animals, but not as a pure alternative. So animal testing is still very much needed in order to sort of test drugs and get new drugs out. Okay, so that was animal testing. We're now going to have a look at clinical trials, which, as I said earlier, can be broken down into a few different phases. The first of which is phase 1. And this is when your new drug or placebo is given to a small number of healthy volunteers. If you're not sure what placebo is, have a quick look now. It should be a term that is familiar to you from GCSE. At this point, you have to give it to healthy volunteers as opposed to patients who have your disease you're interested in, because you are checking that the drug works as expected in the human body and it doesn't cause any unexpected side effects. And whilst this is going on, at the same time, scientists will continue looking at the effects of longer term use in animals. So if you imagine whilst you're running this, that volunteer will have been taking the drug for maybe 6 months. You're going to have animals that have been taking it before that, so they're looking, haven't taken it for a few years, so you can check the longer term effects of that. Once we go through phase 1, we then move into phase 2, and this is when you actually use it with patients who have the disease. So if you're looking at a tablet for heart disease, you're actually going to give it to those patients. And at this point, between 100 to 500 patient volunteers are either are given a new drug, and then you're going to have a similar number that are even given a placebo, or they're given the best current treatment. And this is your first chance for scientists and doctors to see how the medicine affects disease in a real person. Your volunteers are going to be closely monitored to find out about dose, to check the effectiveness, and for side effects. And then phase 3 is exactly the same thing, but you're going to use thousands of patients, so 5,000 plus, as opposed to those 100 to 500 initial numbers in phase 2. Now, phase 2 and phase 3 trials are typically carried out under a double-blind trial protocol. Again, this should be familiar to you, but just in case, I'll quickly whiz through it. In a double-blind trial, this is where neither the doctor nor the patient knows whether the patient is getting the new medicine, a control medicine, so something that already exists, or a placebo. And one of the reasons for that is it allows the placebo effect to be properly measured. So placebo effect is when patients appear to respond to a drug simply because they think it's doing them good. So we don't really know how the mind fully affects the body yet. Essentially, a patient could be swallowing a sugar pill, and because they think it's going to make them better, because they think it's the real drug, they do get better. So double-blind trials allow you to monitor that. It's important to know, and it is important that the patients are randomly allocated into groups. So there is no way of knowing, as a doctor or the patient, what group you are in. There are some difficulties with trials and getting a complete data set. Patients could stop taking the medicine for any reason, so then you've got an incomplete data there. Some don't take it regularly, so they might have to take 2 tablets a day, but for whatever reason they don't. So that can affect your data set. And it could also be that your new drug is so successful that you have to stop your trial because it's no longer ethical to deny your placebo patients that treatment. So it could be, you're starting to see some results after 6 months that are really, really promising. You can't keep putting people in the placebo trial because it's not ethical to deny them a treatment that you know to be so effective. Okay, that was a really, really quick whiz through. Hopefully you've understood all of that. If you've got any questions, post them on the discussion board in Schoology. I'm more than happy to help. And as soon as you're ready, please move on to your next task.